In this video, I want to draw out an action potential. We have a graph here we can use to understand the action potentials. What I will do as I color code them, I'll discuss what is occurring there, just so we can see it graphically besides as we've drawn it out before. So understanding what's going on here is quite important. Now, I'm going to do one thing just to kind of keep us at a little idea here. I'm going to draw a line across for our threshold level. And this is the level that we've got to get across in order to have an action potential actually occur. Now, if you remember, the resting state of a neuron is at negative 70 millivolts. This is maintained by the opening of sodium-potassium ATPase pumps, which maintain the resting potential. So resting potential will continue on until you have a release of a neurotransmitter, will bind to the postsynaptic cell. Neurotransmitters, when they bind, we begin to form this, this nice little graded potential. And as the local event continues on, graded potentials will be anywhere in the 60s. So as we go above negative 70s, we continue rising. We're at 69, heading all the way up to 60. Now, if we're able to get over threshold, we can shoot all the way up to plus 30. Now, here we are at full depolarization, and that is the result of the opening of sodium gates. Anything from negative 69 to 60 up to positive 30 results in depolarization, the cell becoming more positive. This is a result of sodium gates opening. So I'm going to draw a black line here to stop us from, that is everything the sodium gates are responsible for. Once we are done, the action potential has fired, we can continue on with something else. We need to get this cell back down. Potassium gates were open, and this will result in the cell becoming more negative. And as it happens, we have a rapid depolarization event that will happen. Now, this may stop at different levels as we dip down, and level back out at negative 70 as back to resting. Remember, negative 70, by the time we dip at this point, sodium-potassium pumps will kick in to reset everything as due to hyperpolarization. Hyperpolarization events happen somewhere about negative 40. But we drew a little bit lower down. But this will give you a pretty decent idea of what's happening during the action potential. Resting potential maintained by sodium-potassium pumps. The graded potential forming during threshold in orange. As we reach negative or over threshold and we shoot in an action potential, fires to plus 30. So sodium gates then closes due to enzymes will break off neurotransmitters. Then we will begin to further re-negatize. The cell will become more negative again. When we open up the potassium gates, it drops down very low, and then sodium-potassium pumps kick on right here. Reset transmembrane potential back to negative 70 millivolts, and we are back to resting, ready for another action potential. I hope this helps you understand action potentials and what is occurring in a neuron, and this concludes my video on action potentials.